For Chapter 9, we are going to learn about the structure of DNA. And here you can see an illustration of a DNA double helix, something that's quite recognizable to anyone, not just those with a biology or a genetics background. But it's really critical that you think carefully about the structure of DNA that I'll describe in this chapter, because in Chapter 10, we take a very deep dive into how DNA is replicated. And to really master DNA replication, it's critical that you have a strong foundation in the structure and organization of DNA. And so the chapter starts out, and I'm starting out with a pre-DNA concept. So basically, before we knew that DNA was the material that is passed from one generation to the next, carrying with it important information, it was not clear exactly what that material was. Now, scientists were aware of the fact that something seemed to pass from one generation to the next. That's why parents look like their offspring. That is why Mendel's experiments were able to be done and conclusions drawn. But what wasn't understood at this time is exactly what that material is. What is this, the essence of this stuff that is passed from one generation to the next? We now know that to be DNA, but I'm going to spend a few minutes here talking about how we got to that understanding. And the understanding grew from an awareness that this material, whatever it is, has to be able to be replicated, and it must somehow store information that can then be used. Okay, so the fact that I have brown eyes, like both of my parents, I received somehow information on how to produce a brown iris from my parents. So my parents needed to replicate that information. It needs to be retained in a faithful fashion and passed on and used. And the last important point is that for this material that we now know to be DNA, there has to be some mechanism whereby variation can be introduced. We now know this to come in the form of mutations, but it was critical that there be some way that these heritable characteristics could be changed through some unknown process. So it needs to be changeable. So this is an information flow diagram that starts with DNA and takes and illustrates the role of DNA and what is done with the information there, how it's extracted, how it is turned into something, and what the end product is. And so today we're focusing on the structure of DNA, or in this lecture. In Chapter 10, we're going to talk about DNA replication, but then we're going to spend a few lectures talking about the rest of this flow chart. Because DNA is used in a number of ways, this illustrates one of those, and that is the expression of the information that is contained in that DNA. Okay? So in order to take that allele that my parents gave me that produces brown eye color, I have to take the genetic information they gave me, my DNA. That information has to be transcribed and then translated, and a protein is produced, therefore giving me brown eyes. Okay? Now, this is how we now understand our genetic material to be used. But again, we're backing up, starting at the beginning, some of these earliest experiments. How exactly did we get to this point? So, the first experiment that we're going to touch on is one run by a gentleman named Griffith. And what Griffith was working with was two strains of a pathogen, that one of which, when injected into a mouse, would kill the mouse. This is a virulent strain of pathogen. Okay? It infects and kills. There was another strain of this pathogen that when injected to the mouse had no effect on the mouse. Okay, so these pathogens, one of them had a lethal effect on the mouse, the other did not. But what was interesting that Griffith did, is he took those virulent strains and he heat killed them. So if you take that pathogen and boil it, basically, you kill the pathogen. Okay, you've done nothing other than kill it and inject it into the mouse. Okay, and when you do that, the mouse survives. So that pathogen needs to be living in order to infect and affect the death of that mouse. Okay. Now, the critical experiment that Griffith performed was combining living avirulent pathogens, so the ones that don't kill, with some heat-killed virulent pathogen. So he kind of took a little bit of each culture, one of which he had boiled and killed, the other was living, but even when alive, it does nothing to the mouse. Mixed them and injected them into the mouse and observed that the mice actually died. So this is crazy. This is the genetic equivalent of 1 + 2 = 4. All right. And so what he had to do was try to figure out what was going on. And when he analyzed the tissue, he found living virulent pathogens. Okay. And this made very clear to Griffith that the information that was in those heat-killed virulent 3S pathogens was somehow transferred to the avirulent 2R strain, converting them to a virulent form that then killed the mouse. Now, this probably doesn't surprise you much given what we covered in the previous chapter with the movement of information between bacteria. But this was a critical observation in our progress towards understanding what genetic material really is. And so what he showed here is that this virulent information could be transmitted from one cell to another, even if one of those cells is dead. That this could result in transformation of these avirulent cells. But what he didn't know is, what is this material or essence or information? What is its shape, its form within those heat-killed cells that it's able to move over to the avirulent cells? And there was a strong suspicion in the scientific community that within these cells, there were really only two possibilities, one of which was protein and the other being DNA. Okay? They knew that these things existed. They knew protein existed. They knew DNA existed. They knew RNA existed. All of these things had been documented and observed and somewhat characterized, but they didn't know exactly what DNA looks like. They didn't know exactly what was in DNA, and they didn't know how any of these things could potentially carry information that could be passed from one generation to the next, or one cell to the next. And so it was later experiments that clarified this. So we're going to cover two. This first one by Avery, McLeod, and McCarty took this very same virulent 3S strain and spun down some liquid culture. We talked about how these pathogens, these bacteria, could be grown in liquid culture, spin it down, and they just cook it for a bit to kill them, okay, just like Griffith had. Then they homogenize the cells, and they just get basically all of the essence out of these cells, none of the liquid broth that was in the tube. They got rid of all that. So they took all the stuff that you basically get from these cells, carbohydrates, lipids, proteins, all that stuff, and got rid of most of that. They know these things, the carbohydrates, lipids, proteins, all of those things. They wanted to get rid of what they knew wasn't a contributor, and then test for the possibilities of the influence of protein, DNA, and RNA. Okay? And so once they had made this homogenized liquid. They proceeded to test, okay? And we're going to start from right to left, okay? The first thing they did was take these 2R cells. Remember, those are the avirulent cells that don't cause any death in the mice, don't cause any problems. And the 3S filtrate, so this is the stuff from the heat-killed virulent forms. And they sought to see whether transformation occurred, whether you end up with 3S cells. And when you just put those two things together, sure as hell, just like Griffith observed, you get active 3S cells, okay? So the next step they did is they took some of that same 2R cells, 3S filtrate, okay? But they added protease to this mix. Now we're on to the second column from the right. And what protease does is it destroys protein. So we have in this mix, we have living avirulent, no problem, 2R cells. They don't kill the mice. We have the filtrate that we know transformed the 2R cells in the previous flask, but we've removed from that filtrate all the protein. And guess what? Lo and behold, transformation occurs again. You end up with virulent 3S cells. Took some of that treated filtrate that has all the protein removed and treated it with RNase to get rid of RNA. So now protein's gone, RNA's gone, and in this third from the right column, you will see that we still get transformation, even though all the carbohydrates, all the lipids, all the protein, all the RNA has been removed, we still get transformation. Took some of that fluid, treated it with DNase to remove DNA, destroy DNA, and finally they observed that no transformation occurred when they mixed the 2R avirulent cells with this filtrate of the virulent form that was heat killed, lipids removed, carbohydrates removed, protein removed, RNA removed, and DNA removed. Finally, no transformation. Therefore, they concluded that DNA is actually the active factor that carries this information on virulence and could transform the 2R cells. Now, this experiment was substantiated by research, the Hershey-Chase experiment, in which they sought to confirm these observations, this dichotomy between the effect of protein and DNA, using bacteriophages, much as we discussed them in the previous chapter. So what they did is they knew that these bacteriophages, viruses, are basically two-part systems. They've got DNA inside them, and their body's made of protein. Okay? And they know that these things attach to cells, and they put something in the cell that changes the cell. And whatever it is that they put in the cell that makes the changes, that's got to be that material that carries all of this information. So they set out to determine whether what these phage stick into the cells is DNA or protein. And they came up with a very interesting experiment that allowed them to do this. And so in doing this, they labeled the raw materials put into the broth with these bacteriophage with radioactive isotopes, okay? And so they grew bacteriophage in flasks, so I'll just go left and right, that in addition to the normal isotope of P and S, they included some subset or some portion of radioactive isotopes of those two elements. And they knew that each of these, so the S is incorporated into most proteins, okay? And so all of the protein that was created in new bacteriophage would have some portion of their S as radioactively labeled forms. And then in the flask on the left, the bacteriophage that grew in there, because this element, P, is part of DNA, that the DNA that is created in all of these bacteriophage would have some portion of their P within their DNA be radioactively labeled. Okay? And so they took these radioactively labeled bacteriophage, two different ones. One on the left has the DNA radioactively labeled. The one on the right has their S radioactively labeled. And they just put them into a regular culture of bacteria. And in this culture, these bacteriophage are going to infect the cells, and just like bacteriophage work, they're going to attach to the cells and inject into them whatever it is they use to take over those cells with their own genetic information. The Hershey and Chase didn't know whether that was DNA or protein, and so they would be able to determine which it was based on the results of their observation of the cells that we will see here in the next step. Okay, so what they found is that in the portion of the experiment on the right, the phage that had radioactively labeled phosphorus, P32, they injected their radioactively labeled DNA into the cell, and the phage ghosts on the outside did not retain any radioactive signature. So when Hershey and Chase looked at the results of their experiment, they found fluid with a lot of no radioactive signature, and they found within each of their cells lots of radioactivity because the stuff that had been put into those cells was radioactively labeled, and they knew in that particular treatment that what had been radioactively labeled was the DNA, because phosphorus is a portion of DNA, as we'll get to in a moment. On the right, when sulfur S35 isotopes were incorporated into the mix and into the bodies of the phage, they did not have radioactively labeled DNA. And what Hershey and Chase found is that when they looked at the cellular composition of this flask, they found that the cells had no radioactivity in there, but the liquid around them was full of radioactivity because it was full of these empty phage ghosts. They'd stuck their genetic material into the cells, but that material wasn't labeled. The only thing that was labeled was protein. So this helped Hershey and Chase document the difference, or they clearly conclude, they allowed them to clearly conclude that DNA is the material that is inherited and carries this information from one generation to the next. These findings were substantiated by the observation that there is a particular range of the UV spectrum in which genetic information seems to be changed or mutated. So at high levels of exposure to a very narrow range of UV light, that genetic information gets changed. And so if there's the question of whether that genetic information is DNA or protein, the observation that protein absorbs UV light at a much higher range in the spectrum, and nucleic acids absorb UV light right in the range where we observe the effects of UV light on genetic information. This is part of the reason you're supposed to wear sunscreen, right? So you don't get skin cancer, because UV light is directly absorbed by your DNA and it induces mutation. And we'll be talking about exactly how that happens later in the semester. Okay, so when we speak of DNA or RNA, we're actually speaking of a pretty complex molecule that has two primary components. And those two primary components can be broken down into the base and the sugar, okay? And the base is where these individual bits differ from one another, okay? The sugars between different nucleotides, a term we'll get to in a moment, the sugars are all the same within DNA. But what's not the same is the bases. So there are different bases, and you've learned these as guanine, adenine, thymine, and cytosine within DNA. And then within RNA, instead of having thymine, you have uracil. So these bases are classified as being either pyrimidines or purines. And you can see here the difference between a pyrimidine and a purine. Now, I don't want to get into chemistry too much. We're not going to dive into these all that much, but you should know which bases are pyrimidines and which bases are purines, okay? I'm not going to give you a drawing of one and ask you to identify which it is, but I expect you to know which are in DNA and which are in RNA, and which are pyrimidines and which are purines, okay? Now, the other portion of a nucleotide is the sugar, okay? And there are two different forms in which these sugars can occur. They can be a ribose or a deoxyribose sugar, and they are very similar, differing only in this one group at the bottom right of each of these molecules, where riboses have a hydroxyl group and deoxyribose lacks that oxygen, so deoxy, okay? It's been deoxygenated, or it does not have that oxygen. So it's really important that you understand the difference between ribose and deoxyribose, because as you noted in the previous slide, or previous slide, some of these, for example, guanine, some of the nucleotides occur in both DNA and RNA, okay? So if I show you a sequence of nucleotides and it's GGC, you can't tell me whether it's DNA or RNA unless you're able to see what's hanging off of this end of the pentose sugar. I say pentose because it's a 5-point sugar, so you'll occasionally hear me refer to the pentose sugar. And the difference between these two is the ribose is the sugar group of an RNA molecule, and the deoxyribose is the sugar group found in DNA molecules. Okay. Now, we will most commonly be observing these in their forms where they're fused together. And infrequently, we will see nucleosides. This may actually be the last time you see a nucleoside, okay? Just the sugar and the base, because we are most often interested in the nucleotide, the sugar and the base, and also this group of phosphates hanging off the corner here. And so this is a single phosphate group sticking off of this molecule of DNA, but there could be 2 or even 3 phosphate groups hanging off of this molecule. For the most part, we are going to be seeing molecules that have either 3 or 1, and I'll explain why here in a moment, okay? So the nucleotide triphosphate on the right illustrates a few important things that I want you to pay attention to. The first is, I haven't yet talked about any of these parenthetical numbers on the sugar groups, okay? And you can see on this sugar that every one of the carbons has been numbered from right to left, okay, in a clockwise fashion. You can see there's a 1' carbon, so that little hash mark next to the 1, that's the 1' carbon, and the 1' carbon is always where the base is attached. The 2' carbon is where you will find either the OH or the H group, so the making it deoxyribose or ribonucleic acid. Okay, this having an oxygen is RNA. Okay. The 3' carbon is the next one in line. You can see here it also has a hydroxyl group. The 3' carbon is a very important one. You are going to pay a lot of attention to this 3' carbon. The 4' carbon is not numbered here, but it's the next one in sequence. And then you will see the 5' carbon sticking up off this pentagon. And it is to the 5' carbon that these 3 phosphate groups are attached. The most important carbons for you are the 5' and the 3', and that's going to become very clear here in a moment. They're critical for you understanding the structure of DNA, but they are even more critical for you understanding how DNA is replicated, how transcription happens, and how translation happens. It's super important to pay attention to these 5s and 3s, okay? And if you don't, you're going to have a lot of problems. Okay, so here's an illustration of 2 molecules connected to one another, okay? And this is the form we're getting into higher and higher hierarchical structures of our DNA. And what we have here are 2 molecules connected to one another. And what is between them is one of those phosphate groups. So you'll remember on the previous slide, there were 3 phosphate groups. But now that they are connected, there is but one. And that is in part because the 2 that are gone now are what powered the reaction to connect these 2. All right? Now, we are not talking about how that works in this chapter. We will get to that in the next chapter. But it's critical that you understand how these things are connected to one another. And it is here that you can see why I highlighted the 5' and the 3' carbon. Because it is the phosphate group that is attached to the 5' carbon of this guanine molecule. That phosphate group is connected to the 3' carbon of the next sugar in line, okay, from this thymine. And so it is this molecule attached to the guanine base that brought with it the phosphates, used 2 of them to power the connection of this molecule to the thymine above it. All right? So we are looking at 2 molecules that are connected to one another here, and it is long chains of this that create what we call the backbone of a DNA strand or an RNA strand. Okay? And so this is a very simplified illustration of the connection of phosphates and the sugars, which are not really illustrated here, and the base that is attached to each of those sugars. And so what comprises the backbone of a strand of nucleic acids is phosphate, then sugar, then phosphate, then sugar, then phosphate, then sugar. That is what we call the backbone of a strand of nucleic acids, okay? Now, what's in this DNA? Well, much of what we know of what's in the DNA and how it's organized is based on the work of 4 folks that I'm going to talk about here in succession. The first is this fellow, Erwin Chargaff, that was a Jewish man that was displaced by Nazis, came to the United States and was a professor at Columbia University, and his research is what has helped us understand the ratio of nucleotides to one another, or bases rather. He observed that within species, the proportion of A's that existed within their DNA was equivalent to the proportion of T's, and the proportion of G's was equivalent to the proportion of C's. Now, this will make sense when we talk about taking this single strand of nucleic acids that I showed you on the previous slide and pairing it up to its complementary strand when we make this double helix. Remember, our DNA is double-stranded. To this point, I've only shown you single strands. But if you recall in our chapter talking about bacteria, and I showed you those circular chromosomes, remember each of those was double-stranded. How are those strands held together? Well, it's when A's pair with T's and G's pair with C's, and we'll see how that looks in a moment. But we now understand that because of the work that Chargaff had done. And he did this by going and collecting many different tissues from many different organisms. And he didn't know exactly what DNA did. He didn't know its structure, but he knew DNA existed. And what he attempted to do was characterize the occurrence of the different components of DNA, the guanines, the adenines, the thymines, the cytosines. And it was his observation of these frequencies in these different tissues that he collected. And I'll show you here in a minute. But he looked at samples from humans, from oxen, from yeast, from birds, sea urchins, all of these different creatures. And in each of them, he always found basically the same number of T's as A's and the same number of G's as C's. So it's Chargaff's rule that we cite when we note that G's and C's bind to one another and occur at a similar frequency, and A's and T's do the same. Now, he also concluded that it was probably DNA and not protein that was the material of inheritance, because when he looked at the differences between the species he considered, say oxen and humans, he found very different frequencies of A's and T's, G's and C's between species. Okay, most humans were the same in their proportion of A's, and most oxen were the same, but they differed from one another. And what he concluded is that the reason human and oxen are different is because they have different DNA. And it turns out he's right. So here are some of the data from the analyses that he conducted, and all it's showing is that he was able to calculate the molar proportion of each of these bases, G's, A's, T's, and C's, within samples collected from these different species. And I want you to look at the two samples collected from ox, and you can see that the proportion of A and T and G and C, they don't differ depending on the tissue. So the thymus and the spleen have basically the same proportion, okay? Now, when you compare the ox to yeast or birds, then you start to see, holy smokes, look how different birds are, avian, tubercle bacilli, how different those avian samples or bird samples are from the ox. They have far less A's and T's, far fewer A's and T's, far greater proportion of G and C. Okay? Humans have far more A and T than oxen do. All right? So it's these observations that within species, A and T are about the same, G and C are about the same, but between species, they are quite different. That really brought home to him the 2 points I made on the previous slide. Now, if you look down at the base composition from various sources, in the second half of this table, there's something really important I want you to recognize, okay? And it is the ratio of A to T, so in column 5 there, it's always right about 1. Ratio of G to C, always about 1, okay? But look at column 7, okay? There's a big difference between A plus G over C plus T, okay? That ratio should be about 1, and it is. There's a huge difference between that and what is in column 8, which you can't actually see here. But if I add up A plus T over G plus C, is that going to be 1? I want you to think about it. You can do it here because I've put questions like that on exams, and it confuses students because they think, oh yeah, A plus T, A is always equal to T, and G plus C, yeah, G is always equal to C. You put one over the other, of course it's going to be 1. Do the math, see what you think. Okay, so after Chargaff, there was a woman that was studying the structure of DNA. She also made great pioneering efforts in studying the structure of viruses, silicone, graphite, and Cole. But it was her work in DNA that left the largest mark on history. And she was an expert in X-ray crystallography. Unfortunately, this is before we understood the cancer-inducing properties of X-rays. And she died of ovarian cancer at the young age of 37 years old, having already made some incredible discoveries. And it is her image here that was done in her lab of the structure of DNA that really triggered the understanding of how DNA is organized in the two other folks we're going to talk about here, Watson and Crick. Okay? And so these are actually the two individuals that finally published the paper in which DNA structure was definitively explained. Okay? And unlike Rosalind Franklin, who used X-ray crystallography, they used models, much like you might use when you take organic chemistry, so these little ball and stick models, trying to figure out how molecules can go together. So they published the first paper documenting the structure of DNA, and for that they were awarded a Nobel Prize, along with someone who had worked with Dr. Franklin. Dr. Franklin had passed away by that time, and posthumous Nobel Awards are not made. But there's a great deal of controversy about how her information was used, with or without acknowledgement, whether her permission was granted or needed to be granted. There's a long entry on that on the Wikipedia page about Rosalind Franklin, if you care to read it. So lots of drama and intrigue in biology. But at the end, we end up with what we now understand to be the structure of DNA, based primarily on the work of these four people. And so this is where we are left with an understanding that DNA exists in this double helix. Okay? There were some that thought it was a triple helix. This double helix, and there's a lot of data on this slide. Okay? In particular, some very detailed measurements of the distance between major grooves, minor grooves, etc. I don't want you to worry about those distances. I do want you to be aware that major and minor grooves exist. And if you look at this and think about how the DNA is organized, I want you to think about the pink ribbons, if you will, as being the sugar phosphate backbone. That's what those are, and you'll see that in the next slide. The little crossbars, if you think of this as almost like a twisted ladder, those crossbars are the G's and C's paired together, or the A's and T's paired together. And so this is our structure. We are going to talk about major and minor grooves again, but we'll not talk about any of these distances. So here's our structure, zoomed in a bit so you can see the alternating sugars and phosphates along the spine or the backbone of this molecule, and then also the nitrogenous bases, the G's, A's, T's, and C's, that span this distance between the two sugar phosphate backbones of each strand. And those nitrogenous bases come together into a similar place and bond to one another, forming hydrogen bonds that connect them to one another. And you can see those illustrated by these gray or black hash marks between the A's and T's and between the G's and C's. So I want you to note this alternating sugar phosphate organization, and that is based upon what we've already seen here. You can see this phosphate group that is in between the different sugars that are attached to different bases. Remember the organization or placement of these phosphate groups being between the 3' carbon of one sugar and the 5' carbon of the next. And that brings me to the point where I call your attention to the very top and bottom of this illustration, where you can see each of these pink strands, the backbone of these pink strands, is labeled as being either 5' or 3'. And if you follow one of them, so let's look at the very top one that's labeled 5', follow that pink ribbon all the way down to the bottom, and you'll see that it ends in a 3'. Okay? And the other pink sugar phosphate backbone is completely in the opposite direction. The 5's at the bottom, the 3's at the top. And what that means is that these pink strands, these two strands of DNA, are oriented in opposite directions. And we call that orientation anti-parallel. So they are parallel to one another, but running in opposite directions. Imagine driving down Highway 6, okay? The lanes on either side of the median are parallel to one another, but they run in opposite directions. That's exactly what's going on here. So let's zoom in a bit on just two of these nucleotide pairings and look at the sugar phosphate backbone on either side here first. And you'll see on the left, we've got 5' on the top, 3' on the bottom. Look over to the right side, it's inverse. But you'll also notice, look at those pentose sugars. The ones on the right are upside down or inverted relative to those on the left. We have the same structure. They're just oriented or polarized in the opposite direction, okay? And in the connections that bind those two strands to one another, we can see those hydrogen bonds that span the gap between the two strands, holding the G to the C and the A to the T. Now these hydrogen bonds, we're not going to dig into these too much more than this, but these hydrogen bonds, you will always find 3 of them between guanine and cytosine, and 2 of them between adenine and thymine. Okay, so this is the foundational information that I need you to know from chapter 9, that before we move on to understanding how DNA is made and how it's used.